So the capsule is protective. So it's another, you don't really need to have one, but some animals, some bacteria have an advantage if they have one because it gives them protective properties against desiccation, against phagocytosis, gives them adhesive properties. And so we can't really say that any cell needs to have it because in fact there are times when the cells make their capsule and they don't make their capsule. It's just that when they make it, they get additional properties and advantages. Luke?
So for gram-negative bacteria, you said that porins are going to be used to transport antibiotics.
That's the kicker. It depends on the size of the antibiotic. So the porins don't really transport things in. They're just channels. So there's no energy. They're just a channel big enough for things to get through. And if some antibiotics, so sometimes when bacteria are resistant to antibiotics, it's because the genes encoding their porins have mutated so the antibiotics can't get in anymore. So that would be one mechanism for resistance to antibiotics.
So then at that point, do you just have to try and develop new antibiotics or is there any other just relying on the immune system?
Well, in a clinical case, so imagine if a person had a really bad infection, gave them antibiotics and they didn't work. Either the person would get better in the meantime, if it was you, or if it's like some 95-year-old person, it's like, yeah, they would stay sick and then hopefully get a different antibiotic. Because antibiotics are relatively new. Until the 1940s, they didn't even have them. And so for most of human history, you just got better or you died. That's it worked. Sorry. Question. Let's see. Did that answer your question about this, Christian?
Yeah.
Yeah. What? Yeah?
There's a lot of outer coverings.
There is a lot of outer coverings, and they're big and thick. There are a lot of outer coverings. We didn't talk about the cell wall, and you're not going to be responsible for it, because you guys have already learned about cell walls before, and bacteria have cell walls that are similar and have homologs to eukaryotes. This is a little exciting preview that you're going to be reading about inclusions, sulfur granules. Here's a really cool one. Magnetosomes. They're actually membrane-bound inclusions. You guys can learn about that. That's part of the reading assignment. Okay, here's where we're starting today. There's no questions. So today we're going to talk about two things. Flagella and motility is one thing, and spores, endospores. And that's the end of what we're going to get through for the Monday's test. Bacterial flagella. So you guys learned about eukaryotic flagella in cell biology. Bacterial flagella are similar in that they have the same role for the cells that have them, but they're really different in terms of their structure and how they work. So if you were to define them, you would say that they're propellers for bacterial locomotion. So the helpful word in that is propeller. So unlike eukaryotic flagella, bacterial flagella rotate around a central axis like a prop on a boat. This picture in the book shows, unfortunately, it shows some flagella here, but this isn't the cell that the flagella are attached to. This is differentiating between fimbriae, which we talked about last time, which you can barely see in this micrograph, and flagella, which are much, much longer. Flagella can be 15 to 20 micrometers long, which is as much as 20 times the length of the cell. So if this cell had flagella shown on here, they would go all the way over to the wall. So they're super long relative to the size of the cell. They vary in their pattern around the cell, and that is actually useful for identification of bacteria. Different bacteria have their flagella arranged around their cell differently, and that can be used for identification. So here's, oh dang it. Every time you touch your finger to this, it goes way, way, way out. This is an example. This is a species of Pseudomonas that has flagella just on its end, just on one end of the cell. And when that happens, they're called polar flagella, meaning that the flagella are on the pole of the cell. So these are vocabulary words to learn because you're going to use these to describe cells that you see, and also they help with characteristics of identifying cells. The other vocabulary word on this part of the slide is monotrichous, meaning that there's just one cell. Mono meaning one. Trichous refers to having hair, so it has one flagellum on the end of the cell, and that's enough to help it move. Here's an example of one that has lophotrichous flagella. Lopho means tuft, so it has a tuft of flagella all on one end. This is an example. This is Proteus vulgaris. It has, so some, I'll get to that in a second. These guys have their flagella arranged all the way around themselves. Peri meaning around. Peritrichous flagella. So the flagella come off all sides of the cell. So various ways that they can be arranged in the cell are characteristic for different types of cells. So in terms of what all they're used for, they're used generally for swimming, but some kinds of bacteria have other purposes for them. And one of the interesting purposes is sometimes they're used for a sense of touch. Some bacteria have different kinds of behaviors when they're swimming through liquid than when they're on a surface. So some bacteria actually use them to detect surfaces, not to necessarily bind to them, like Fimbriae. It can be used as a sense of touch, in quotation marks. So some bacteria will have flagella, and they'll swim through the liquid, and then when they come up to a surface, which they might want to colonize to make a biofilm, which, remember, that's fair game for the test too. What is a biofilm? Growth of bacteria on the surface. Some bacteria come up against a surface, and the torque on their flagellum as it spins changes when they get to the surface, and they can use that to sense the pressure difference, and they know that they're up against a surface. And when they get to the surface, they change their gene expression, and they do all kinds of different things that they weren't doing before. So let's look at the structure. We're going to talk a little bit about the structure of the flagella, but spend more time. Yep, Ben?
What was the lophotrichous again? Tuft.
Lopho means tuft. So tuft of, usually this figure shows lophotrichous flagella, but oftentimes it says lophotrichous polar flagella because the tuft is on one end. All right, let's look at some flagellar structure. So this figure shows that gram-negative bacteria and gram-positive bacteria have different flagellar structures. They would have to because they have different cell walls for them to go through. You don't need to memorize every single part of this figure. That would not be a good expenditure of your time at this point, but it's a good review because you can see the periplasmic space here in the gram-negatives, the outer membrane. There are something like 20 different proteins or genes. There's 20 to 30 different genes for flagellar proteins depending on what bacteria you're talking about. So there's a lot of complexity in here. The part that goes through the cell wall and membranes, the basal body, if you're going to break this down into just three parts, the basal body has a whole bunch of proteins associated with it. We'll talk about some of them in a little bit. The hook is the attachment part that links the filament to the basal body. So one of the cool things about the flagellum in terms of assembly is that it's built by self-assembly. So we talked about that once before, the idea of self-assembly. Meaning that there's not a machine that synthesizes the flagellum. You could imagine that there would be some kind of a machine that puts the whole thing together. So clearly you can't put the whole thing together and export it into the cell. Why is that? It's enormous. It's way too big for that to happen. It's like 10 times the length of the cell. They probably wouldn't even fit in the cell. And not only that, you couldn't build a scaffold in which you would build it inside there because it's so long. So what happens is that the monomers that make up the filament get transported out through the center of the basal body, out through here, and all the way out to the tip of the flagellum, the growing tip, and it lengthens from the inside out. There's a picture of it right here. It shows the outer membrane. Here's the ribosomes and the mRNA associated with it. The proteins that are encoded by the genes for the flagellum get pumped through here and then out, and it grows longer and longer and longer, and has to get pumped way out to the distal end of the flagellum as it grows. So there's ways that you can shear off flagella and get them to grow back. It's not as if it's super hard to snap these things off. but it's important that there's no machinery to do it. Dan?
So just the filament is built by self-assembly, or the whole basal body and hook too?
Well, the stuff that goes through this part here that goes right through, this probably has some transport apparatus that helps it get out there.
Okay.
But the filament that goes through here is the part that has to get synthesized by self-assembly because it's so far away from the rest of the cell, there's no help for it. So how does the flagellum work? I'm going to skip down to this picture. The flagellum in bacteria works, as I said before, by rotating around in the cell. The entire flagellar filament rotates within the basal body. So that's different from what happens with eukaryotic flagella. Remember, in eukaryotic flagella, you have, remember the cross-section you learned in cell biology? There's 9 tubules, 9 pairs of tubules wrapped around here. There's 2 in the middle. Does that sound familiar? And then, where does the motion come from? Do you remember that? So if this pen is one tubule and this is the other in a pair, they're connected together, Tony. They slide back and forth. It's not actin though. You're on the right track. Yeah, it's the motor maps. Remember the dynein and kinesin? And there's these little guys in here with ATP hydrolysis and it steps along. Remember that? And it changes conformation. In eukaryotes, there's 2 of them together. There's 9 bundles of these and one in the middle, and one slides past the other, and then it goes back like this and whips back and forth. Remember that? In bacteria, the whole thing rotates around like this in the basal body. And the way it moves, so here's a little bit more detailed picture of it. Here's the basal body, the hook. This whole thing has to whip around, and it goes really, really fast. They rotate as fast as 1,000 revolutions per second, which is unbelievable. And when I was remembering about this, I thought, well, it must be per minute, right? That doesn't seem even believable. It's really per second. So it's spinning around there really, really fast. And this part is what spins. The way it is energized is using protons. So there's a place right between these proteins, and you don't have to memorize which ones it is, but it's between the motor proteins, mote A and mote B, and these right here, where proteins go through. So this is referred to as the motor, and that's where the mote name comes from. Just like a real motor, there's a stator around the outside that's stable, and there's a part that rotates in the middle. This part here is the part that rotates. And it rotates based on the power from the protons, the proton gradient that is just outside the plasma membrane. So if you draw this out a little bit farther, there's a whole bunch of protons out here. This is a crappy system. Those are protons. There's a whole bunch of protons out here relative to the inside of the cell, and that is the energy source to get the flagellum to go around like that. So how do those protons get out there? You guys have learned that before. Were you going to say something? No?
Is that the electron transport chain?
Electron transport chain is making a proton gradient, so respiration gets the protons out there. Some bacteria can't respire, and so they can't use the electron transport chain. They use an ATPase to pump them out. It costs them ATP to get the protons out there. That's not to say that ATP is not the energy source. I'm going to write that down. ATP. If I put my hand on this, well, there we go. ATP is not the energy source. In eukaryotic flagella, ATP is the energy force because that's what drives the motor maps to walk along the tubules. The energy source is the proton gradient out here. It causes that thing to whip around, and as it does, it makes the flagellum spin. So what happens with bacteria, any questions about that so far? What happens with actual motility, though, if a bacterium can swim, that's great, but unless it has direction and purpose, all it really is doing is just moving around randomly. And so that's actually not what happens. What happens is the bacteria are usually attracted to something. They're either attracted to something or they're repelled by something. So the kinds of things they're attracted to are things that they either need for food or energy or maybe protection. So when they're attracted to something, that's known as chemotaxis. And this is true in all kinds of cells that move, including white blood cells. It's movement toward a chemical gradient, or chemical attractants, I should say. So typically we think of chemotaxis as some time when the cells are moving toward an attractant. It can also be away from a repellent. So the way that works is that the bacteria, in order to move, rotate their flagella, and when they're going forward, most bacteria rotate their flagellum counterclockwise. So the whole thing moves around like the prop on a boat. And imagine if on the prop of the boat you had a whole bunch of ropes tied to the fins, and then all those ropes were spinning around as well. That's what the flagellum is like. And so when they go counterclockwise, the bacteria moves forward, and then what happens if you want to stop? They don't have a steering mechanism. But if they want to stop, and they don't have brakes, they have to put the flagellum in the opposite direction. So instead of going counterclockwise, they reverse the direction of the flagellum and go clockwise. And that causes the flagella to fly apart, as shown down here. And all of the, in this case, it was a Peritrichus bacteria. And they all flew apart, and then the cell tumbles in space and changes direction. It almost always goes a different direction just because there's so many options. It's random. The change in direction is random. And then it resumes counterclockwise motion again, and the flagella all go in the same way, and it pushes them forward again. So that's shown with a monotrichous flagellum, a peritrichous flagellum. If they're going counterclockwise, they go forward for a certain amount of time, then there's a change in direction of the flagellum, and they stop and reorient. So the way the change of direction works is shown back here. There's switch proteins. These. We'll talk about this when we get to like chapter 14 or something, we'll talk about it. But there's other proteins in the cell that are responding to a signal transduction cascade, a phospholipid cascade. They come in and grab a hold of the switch, and when it grabs the switch, it causes the bacterium to change its flagellar direction to the opposite direction, and then it reorients randomly, and then goes off in a new direction. So that's what's shown right here. So that means that they can move around, but it doesn't actually help them too much. This is just the way that they, this is just the first part of it. In order to be meaningful, chemotaxis is the result of what's called a biased random walk. Biased random walk. And it's described as those movements of the flagellum that I was just talking about result in what are called runs and tumbles. When the flagellum is going counterclockwise and the bacteria moves forward, that's called a run. It's a series of runs and tumbles. This is the jargon of the field. This isn't stuff I made up or the book made up. A series of runs and tumbles. So the runs are the forward movement with respect to the bacteria. That is the counterclockwise direction of the flagella. And the tumbles are the stops. That's the clockwise direction of the flagella. So they run forward, they stop, they randomly reorient themselves, and then they run forward again, and then they stop, randomly reorient themselves. And so that's how they move around, but that would not get you anywhere, right? Unless you have some way to control the amount of time you spend running in different directions. So what happens with chemotaxis is that the cells run for a longer period of time when they're going in a favorable direction. So the runs. So if I write longer, you might assume that I mean longer distance, but it's really a longer period of time, which results in a longer distance, right? So it's better to think of it as frequency of runs. Runs are less frequent when the cells are going in a favorable direction. So that's why this is called a biased random walk. This is the biased part. It's random in terms of the direction the bacteria are going because they don't have the ability to steer themselves. They establish their direction randomly, right? But it's biased because the frequency of the tumbles is determined by the concentration of the material that they're looking for. So this is the biased part. This tumble part is the random part. Stops and changes direction. The direction is the random part. So there's a figure in the book that shows the result of this. It's right here.
Dr. Eglin?
Yeah.
Are they going to be more favorable or more frequent when they're going in a favorable direction? The runs?
Oh, correct.
Yeah.
Runs are less, tumbles are less frequent. Sorry. Good catch. The tumbles, so you don't have frequency of runs. Runs is a default, right? It's the tumbles that are more frequent. When they're going, no, that's right. Tumbles are less frequent. Oh, that's correct. Tumbles are less frequent when going in a favorable direction. Right. And that's because if they're going in a favorable direction, they don't want to change direction. They just want to keep going. So that's shown in this figure with this gradient. Can you guys see how this is greener over here than it is over here? This is meant to show a gradient of greenness, and the greenness is the attractant. It's the place that the bacteria want to go. So imagine that the green stuff is the sugar they want to eat, or maybe it's the oxygen they need. So bacteria can be attracted to chemicals that they want. They can be repelled from chemicals that are toxic. They can be attracted to oxygen or light, all kinds of things. So this greenness represents that. So up here, there's no gradient. And when that happens, the cells move forward, whatever forward is to them, right? They tumble, they move again, they tumble. They don't really get any place. They have pretty much uniform distribution of their tumbles, and consequently, they don't make any progress. In this figure, there's a gradient that they're able to respond to. So they run for a while, they tumble. If they go in a favorable direction, they would go longer, but this guy unfortunately went in a neutral, essentially, direction. So it tumbles again, goes again. This time, fortunately, the front end of the cell, I'll draw it right here. This stupid thing will let me draw. Say this is the cell, and these are the flagella. Fortunately, it's going in a favorable direction that time, towards the gradient, so it spends more time going in a favorable direction before it tumbles again. Eventually, it does tumble, and then depending on how it randomly reorients, it may tumble again pretty soon, or it may not tumble. So in this case, it tumbles again pretty soon. Here it goes very soon because it's not going favorable. Finally, down here, it gets itself going in a favorable direction, and it goes for a longer period of time. So overall, eventually, the cell makes progress towards the direction that it needs to go, towards the favorable direction, right? It doesn't really want to go that way. That's just the favorable direction. So the tumbles are less frequent when it's going in a favorable direction. They're more frequent when it's going in a non-favorable direction, so it doesn't go as far. Does that make sense? So we're going to talk about the molecular mechanism about how that works when we get to the chapter about control and control of cell functions. So, questions about that? Here's what the outcome looks like. This is one of the ways that you can detect chemotaxis in bacteria. And this figure shows a Petri plate. This is a Petri plate that has semi-solid agar that is runny enough that the bacteria can swim through it. Tony?
So is it proper to describe it as saying they run for a longer amount of time?
They run for a longer amount of time, but not necessarily. Yeah, that's proper to say. It's true to say that, but that's not the way the control works initially. See what I mean? So this is a Petri plate with really runny agar in it. It's so runny that if you tip it over, it would dump out, but it's firm enough that the bacteria can't move very well unless they actually have motility. So in this picture, there's an attractant in there, something that the bacteria want to swim towards and eat. It's probably an amino acid or a sugar. And what the people who set this up did was they inoculated 3 strains of bacteria in here. One right here, and one right here. They just took them on their loop, stuck it into that sloppy auger, and then let them grow. And the bacteria that are non-motile, if they can't move, they just sit right here and grow until their nutrients are gone, and then they have to stop growing. These guys are motile. This strain is motile, so it can move around a little bit, but it doesn't get a huge advantage. It just eventually kind of diffuses out because of its motion. But these guys, as they grow, they're chemotactic. So as they grow and they use up the attractant, which is the sugar that's in there, they use up the sugar and they create a gradient themselves. So in the center, the sugar is all used up, and just outside the center, there's a little bit more. And farther away, of course, there's the full concentration of attractant. And so from here to here, from here to here, you have the gradient that we were just looking at here. So there's a lot of stuff out here, a little stuff in here, because the bacteria ate it up and made their own gradient. And that causes them to have directionality and move away from the center. So this kind of a ring, if this is a full Petri plate, that would be a ring with a diameter of about that big. And so just in the course of 24 hours or so, you could get that much movement in the cells. Not an individual cell, of course, because they're growing and dividing, and it'd be like the progeny of that cell are swimming out as they grow.
So in that example, would the flagella ever tumble, or would they just keep running?
They would.
They would randomly tumble.
They would have to tumble from time to time, and that is
From time to
because.
time.
Yep. But if they have a favorable direction, they would spend longer time swimming in the favorable direction. But they have to have a short-term memory, and we'll talk about that another time. They've got a short-term memory so they can compare where they are now to where they just were, so that they can always assess what's favorable. Tony? Do they stop? When they tumble, they do. Is that what you're asking?
No, do they have like just tumble and they don't move anymore? Like if they're eating something?
No, they're eating stuff. Oh, I see what you're saying. Like if it was deer in the field and they stopped to eat the corn?
Yeah.
No, they eat as they swim. That's a good question. So what they're eating is molecules, right? And so the molecules bind to a receptor and it gets taken up into the cell, but they don't have to stop for that to happen. I can see why you'd ask that. That's a good question. Yep, Matt?
Are there only bacteria on the outside then, or is it on the inside?
That's a good question. Why would there even be bacteria still on the inside? It's because the inside eventually here is probably getting all acidified and poisoned, and the bacteria in there probably aren't real viable and they're not swimming. The other thing about swimming is it takes a lot of energy to do it. It's really energy intensive because it takes, and the people who study this know exactly how many protons have to fit into that ring to get it to go. And they also know you can relate the speed of the flagellum to the difference in concentration gradient, the difference in delta psi, remember that, across the membrane. So if there's a really strong concentration gradient, the flagellum goes faster, of course. If there's just a few extra protons, they get in there and eventually it's enough to make it go around. And so these guys that are still in the middle are ones that got left behind because they didn't have enough energy to move, or maybe they even got poisoned by all the acid that got made. Different kinds of bacteria give you different patterns, and depending on what kind of organ, so look at, let's see, here, this one go. This one has a different situation where the cells are somehow staying associated with each other. So you can imagine a scenario where they make rings and they don't really get left behind very easily. And depending on what they're growing, they have secondary metabolites that can get really, really complicated. We're not going to talk about this right now because I can tell that you're kind of saturated with this much. But this is basically the opposite of chemo attractant. It's repelling. And it shows how you can have rings of cells running away from something like ethanol. E. coli is negatively attracted by ethanol, and so they try to get away from it. Questions about Libby?
Well, for those, I can see how having flagella would be very beneficial, but what do non-motile organisms, do they have extra, they're just lucky if food comes their way?
Yeah.
Or
So if you think,
pellets, or do they have defense mechanisms? Maybe that's what.
they would have defense mechanisms. So an example of a place where you would find non-motile bacteria would be in places where they're stuck in a biofilm. Because if cells are in a biofilm, in fact, if motobacteria join a biofilm, they often lose their flagella and their motility. And so if they're in a biofilm, they probably aren't swimming all around looking for their food. They're stuck to a surface, but what would be the nature of the environment? The nature of the environment, we'll talk more about this later, is that there's stuff flowing by them. So imagine the bacteria that are stuck to your teeth. This is awesome. The bacteria that stuck to your teeth are largely non-motile bacteria. There's just a few that I can even think of that are motile. They stick there, and they're always bathed in saliva that flows by them. And they can just take the stuff out of the food that goes by them. They don't have to swim all around. Just take it out of the environment because there's flow past them.
Is it kind of like a filter feeder?
Yeah, it would be. Like the biofilm? You mean like in an aquatic system? So the filter feeders would be, well, the kicker, it would be, but what's coming in is coming in a molecule at a time, right? It doesn't have, so what's an example of a filter feeder? Like a clam or something? So they're able to take up huge volumes and filter it through them.
There's
Whereas the bacteria, what would be another example?
baleen whales.
Oh, yeah. So they're taking up stuff that is dissolved, and they're taking up a bunch of water with it, whereas the cells are using transmembrane proteins as transporters to bring the stuff in. But if they're bound to a surface, like a biofilm, you could kind of say that because they'd be bringing stuff in and then making some kind of waste product that's filtering through them. Kind of like that. I hadn't thought about that before. Let's see, other questions about, does everybody get why there's a gradient here? Where's the gradient come from? The bacteria made it because they ate all the sugar, and now there's a gradient because they ate up all the stuff in the middle, and now they swim away to get to more. Ben?
Would it be possible to set up an auger that had a sugar gradient built into it? So like you put the chemotactic bacteria on the side of the cell with the least, or the side of the auger with the least sugar in it, and observe to see if they move towards the.
Yeah, you can totally do that. You could, there's a couple different ways you could do it. One way is you can take the auger and take a pipette tip and turn it upside down and use those little cork bore and then shove it in there and then pull it out, and then you can put your stuff inside that little hole. Another way that you can do it is, you'll be sorry you asked. This might be fun. How could we figure that out? Another thing you could do is do it in a test tube and put your auger in there and then put your attractant on top, and then it diffuses down and you have a gradient like this. And then you can put the bacteria throughout the tube. This is kind of like what you guys are doing in lab, only it's more like a vertical version of this. But you could create a situation where diffusion makes your gradient too. Another way that people sometimes do it is they take their Petri plate and they put it on the side when they pour it, so that you pour your stuff that contains attractant down here, and then you set it square like this after that's solidified, and then you pour plain media that doesn't have bacteria in it in there, or it doesn't have attractant. See what I mean? And you make a kind of a wedge-shaped thing, and then there's way more down here than there is over here. That would work.
Are you trying to separate bacteria in terms of their speed designs? Like you're going to put them on one side and see how fast they get there, or what's the point of?
No, this was ways to create the gradient.
Yeah, I get that.
Yeah.
What is that do for you?
Well, what did you have in mind for doing that?
I don't know. I was just wondering.
It could theoretically be.
Yeah.
Well, what you could do is, so what you said, Christian, is true. You could theoretically separate out those that are motile from those that aren't. Imagine if you wanted to, so I knew some people were trying to find mutants in the chemotaxis machinery so they could figure out how cells responded to these particular organic pollutants that were in the environment, because they were thinking if they could get bacteria to swim to the pollutants, then they could eat them up. And so they did experiments where they mutagenized the cells and then sorted out those that were unable to detect the attractant anymore. So that'd be the kind of thing you could use that for. It'd be a way to select for mutants. A little off track there. Questions about chemotaxis in general? Runs and tumbles? Tony?
So, in the case of the gradient, how is bacteria, how's their food usually distributed?
So in the environment, if it's a place where there's any kind of motion at all, it would be distributed uniformly, because imagine if you're talking about a lake, the nutrients are homogeneous if there's any kind of movement in the water at all. That would be, it depends on the environment you're in. If they're being produced by a particular source, so in the environment, bacteria are eating something that was made by something else, right? And so there might be a place in the environment where one organism is creating substrate for another, and so it's localized and it's diffusing away from the source at which it's being made. You can imagine that too if it's not just depends on what kind of environment you're in. Other questions about chemotaxis? I hate to really pour it on the day before the exam because that's kind of rude. Let's say just a little bit about spores. So, the second thing we're going to talk about today, besides flagella and motility, was bacterial endospores. The book has a whole bunch of stuff in there about endospores and formation, and that's probably because it is really, really well understood. And it is actually, the formation of bacterial endospores is one of the early models for developmental biology and how cells can change from one type to another within the course of development. So in terms of, let's back up a second. What is an endospore? Typically we call them spores rather than endospores. But what they are is heat-resistant structures, whoops, in the right heat. They're used for survival. They're not used for reproduction. So if you learned about spores in some plant class or Bio 120 when you were talking about fungi or something, in that case the spores are for reproduction. Like spores from a mushroom, those are reproductive structures. Spores from bacteria are survival structures. And so they're resistant to heat, not just heat, they're also resistant to UV, they're resistant to desiccation. That doesn't look right. Desiccation, they're resistant to chemicals, all kinds of things, because the cells actually make this structure that is dormant, and they have layers of material around the outside, and that's shown over here. Inside is the core, and that's where the cell's genome is. And then there's many layers of protein, and not just protein, but some other chemicals that make them super resistant. There's a figure in your book that shows, well, first let's look at this. One of the, in terms of what you guys are going to see this semester, one of the important things about spores is that different bacteria have different characteristics in terms of how their endospores look. And it depends on, or that they vary with respect to the position of the spores inside of the cell. So here's an example of these are not like earth-shatteringly difficult vocabulary words here. This shows one that's in the central part of the cell, one that's terminal, one that's not quite terminal, so it's called subterminal. Sometimes they make these tennis racket-looking things because the endospore swells up the end of the cell and so it gets bigger than the regular diameter of the cell and gives you a swollen sporangium. This structure around the spore is what's left of the old cell, and that's called the sporangium. So I'm just going to show you, you are not responsible for all the steps of this figure. I just want to show you one thing about it in terms of how spores develop. What happens with spores is they go through, the sporulating cell begins going through cell division, and what eventually happens is you get asymmetric division of the cells. So you have, rather than two cells that are uniformly spaced out, you have one that's bigger than the other, and then the big one, the big one actually engulfs the smaller one, and the genome in the bigger one gets broken down, and the layers contribute to the formation of the endospore. So you don't get a new, you don't end up, here you go at the end, you have one endospore that resulted from cell division. And so rather than getting two new cells, you get one endospore, which seems like kind of a ripoff. But bear in mind that the endospore can then lay dormant in the soil for 100 years or wherever it is and allow the cell to come back when the environmental conditions get better. So this is triggered by starvation and desiccation, and there's a whole big, again, phospholipid single transduction cascade we'll talk about another day. And the result is the formation of this resistant structure that hangs out for years and years and then can germinate and make new vegetative cells again. Tony, what were you going to say?
Are there 2 genomes in this one?
Nope, there's just one genome in the endospore. Because the second one, and I mean, these are essentially equivalent genomes, right? They're daughter cells. One of them gets degraded as this step happens. The spore gets engulfed and one of the 2 genomes gets broken down. So we're not going to take I mean, we could talk a whole day about spores. And that's not going to happen. Questions about? So what are some examples of bacteria that make endospores? Ones that you would care about. Bacillus anthracis. So you guys were probably not even born when the anthrax attacks happened, right? Were you? No? It was so in, let's see here. It was 2001. Some guy out at the CDC or something picked up a whole bunch of spores. Were you born in 2001?
Yeah.
Yeah.
How old are you? Oh, that's early.
97.
Oh, 2001, that's when my kids, okay, never mind, I got you. Do you guys remember that? Do you remember when the anthrax attacks happened? Do you remember whose office it happened in? It was a senator from South Dakota. It was Tom Daschle's office. It was our South Dakota senator
Oh, yeah.
whose office received a package in the mail full of anthrax spores. And one of his interns opened it up, who happened to be an Augustana student working in his office, opened up the envelope and out dumps a bunch of white powder containing bacterial endospores. And so that was in 2001. It was a domestic terror attack. And several people died, and senators in the Washington, D.C. area received mail, and some
Yeah.
media, some news media places got some mail, and then they had to shut down and sanitize the office building. And it was a big deal to sanitize it to kill all the spores because they're resistant to chemicals and heat, and it's really hard to kill them. So in order to kill off the anthrax spores in the Senate office building, they had to seal it up and raise the humidity because they're resistant to desiccation. If they're dry, they're fine, but if you can get them nice and humid, and then they treated them with basically gas-phase Clorox as an oxidizing agent, chlorine gas, and if the humidity's high enough, you can kill them that way. And until they got that figured out, all of the mail that was sent in the Washington, D.C. area was irradiated. And my wife and I lived out there at the time, so I have a very good memory of the mail that came to our house being brittle, as if it had laid, if you leave the newspaper on your driveway while you're on vacation, it's all brown and brittle when you come back. All of our mail looked like that when it came to our house because it had gone through radiation in order to kill off any potential spores that might be in the stuff that was delivered to us.
Wow.
So that's one example of an organism. Causes anthrax. Another one that you would know, Clostridium botulinum. Causes botulism. And when people get botulism, most of the time when people get botulism, if it's adults, it's because they've eaten food that was canned inappropriately, and the vegetables that they picked and canned at home had spores on them, and because spores are resistant to heating, they often don't get killed in the process, or they potentially could not get killed in the processing if you don't do it right. And they can germinate and grow into vegetative Clostridium botulinum. And then they make toxin and gives everybody botulism, which is a big deal. We'll talk more about that when we talk about diseases. And we'll talk about some other examples of spore-forming bacteria.
So the autoclave won't kill spores?
If you do it long enough. That's why we autoclave, because it has increased, So boiling doesn't necessarily kill them, but the autoclave combines pressure and moisture and heat. High heat, 121 degrees Celsius plus steam, moist heat, is what kills them.
Was that art student, was she okay?
Yeah, she was okay. Thanks for asking. You're the first person who's ever asked that. Some people died, though.
I just hold on to the still thinking about her.
You're worried about her. Yeah, the government department and the history department can tell you all about her. I can't remember her name. That was before I was here. 3 years before I came. All right, any questions about the exam anything about today's content, Ben?
Well, this isn't about today's, but
That's fine.
do we need to know anything about bacteriophages?
Is you would need to, we're going to talk about bacteria phages. So anything that you need to know about them is true of other viruses as well. And we're going to talk about them totally separate when we talk about applied uses of them and exchange of genetic material. Other questions?
So you said we weren't like necessarily responsible for cell walls, just about the things that we've talked about in our notes so far up to this point.
Cell walls?
Isn't that what you said a little while ago?
Yeah.
At the beginning of class, I thought you said not responsible.
That you're not responsible for cell walls? What was the context to that?
I think,
You're responsible for the membrane and the cell wall.
Okay.
What was I talking about?
I think you said different types of bacteria found within the cell walls, because you pulled up like a picture.
Might have been a different cell wall.
It keep going up. It was like a table
No, that was
right there.
cytoskeletal elements.
Okay.
Did I call them cell wall? I
Yeah.
was thinking about the cell wall. You're not responsible for the cytoskeletal elements. You're absolutely responsible for the cell wall. And in fact, how would you draw the cell wall? You wouldn't draw it like this. Please, whatever you do, don't just go like this. That's not sufficient. Right? We talked about this.
Good.
This is the level of detail we're talking about here. Other questions?
What about for stuff like the herpes virus and polio? Do we like those figures that you pulled up that would explain
Yeah.
how they work?
And the reason we talked about those is because those were examples of RNA viruses. And we're talking about what has to happen with different kinds of RNA viruses, with their positive single strand, negative single strand. So those are the examples the book gives of, like for example, influenza virus is a negative single-stranded RNA virus.
But do we have to know how those figures that you showed us about how they work and everything, those in particular for herpes and polio, or just like those types of viruses in general?
Yeah, those particular viruses are just illustrative of the group that they're part of. Yeah.
Sounds good.
Does that answer your question? Yeah. Anybody else? Go to inauguration. You will have a hard time finding any faculty or anybody around after 2:45. So if you have questions today or over the weekend, be sure to email them. Yeah.
Joe.